Thank you very much, everybody, for the invitation. It's actually quite nice to be back. Actually, I postdoced here at the University of Toronto, just down the street. So it's coming a little bit full circle for me. I was asked to give you a little bit about Moderna as a company, and then also to talk about future job prospects and what that would look like, recognizing, of course, that we're mRNA-focused company. So for about 10 minutes, I'm just going to give you a little bit of a backstory of who Moderna is, because I think we're all familiar with the name, but I don't necessarily think Moderna has had a chance to really tell everybody what they're really about. And that's where we're coming out of this pandemic mode. We're finally able to go. I mean, the reality is most people wouldn't know this, but for the first year of the pandemic, when doses started coming into Canada, Moderna Canada was 4 people. It was not this giant pharma company that had a lot of know-how of how things were working. It was, in fact, a small biotech company based out of Boston that just happened to have its technology ready to go at the right time. And I have no doubt there will be movies made about this. There are already books about this. But the chance to actually have had an opportunity to join at that onset and then to not just work through a pandemic, but then also build that company to the point where I've now been there for 3 years and grown it from 4 people. Now we're up to over 20 that are working here in Canada, based for the Canadian organization, but then there's another 20 or so that are actually working for Moderna, based in Canada, but actually working for example, its Boston hub. Okay, so as we continue to grow, there is a larger vision for where this will go. And my hope is that you're going to, at least in the next 10 minutes, get a better sense of what that means. From there, we'll transition into, so what does that mean for you, right? And how could this potentially be something that you may want to look at for future career opportunities, whether that be at Moderna. But I think when I was preparing this talk, one of the things that I felt most excited about is I'm looking at a room of individuals who are going to enter a workspace at an unprecedented time when it comes to data and what it means and how we're going to use it into the future. And I think you guys are just so well positioned to not just walk into that, but to actually teach the rest of the world how we're going to do it. So I envy you. I really do. I think you're going to be entering the workspace at just the most incredible time in generations. So let's see what that could mean, at least from a mRNA perspective. So I'll start off with just a very basic overview of what Moderna looks like now. And I'll focus on just 2 key numbers here. The first is the number of employees. When I started 3 years ago, there was about 1,000 people at that point. It has now grown to over 5,000. By end of year, we'll probably have about 6,000 people. That's a massive amount of growth for any organization to go through in about 2.5 to 3 years time span. We've gone from a Boston-based company to a company now that with a global footprint, with countries operating all around the world. Sorry, I missed my mark. I was supposed to be here. To now setting up operations elsewhere, especially in the Asia-Pacific region. We have offices in Australia, we operate in the Middle East, and now we actually are starting to look at, and I'll get into this a little bit, how we're going to build out into Africa, for example. The second number is actually on the top right, which is the number of programs in development. 3 years ago, that number was about 9. Now that we are at the stage that we're at, and we've recognized what the technology can potentially do, we are at about 47 programs in development, over 30 of which are already in clinical trials. So I will give you a little bit of an overview as to what the thinking is and how we can do that so quickly. So we'll take a step back because not everybody really knows mRNA, so I'm just going to level set what mRNA really is. For the first thing to really understand is that mRNA just didn't pop up, as evidenced by the fact that a Nobel Prize was recently given. mRNA also, to give credit, there have been decades worth of actual development that has happened behind that science. And in fact, we good? Okay. I'm just making sure that they can hear online, so. Fair enough. Okay, so basically, for Moderna, it's actually been about 10 years. It's a 10-year-old company. And in fact, there has been a lot of development behind the scenes, most of which isn't really actually known, although it is actually scattered throughout the literature, but nobody reads very obscure journals on lipids and things like that and would take the time to actually pull it all together into one story. But the first thing to really understand is actually there is a lot of science that has actually been done around this technology. The next thing is, well, how do you actually look at mRNA? And mRNA is actually something that's quite different to us, as opposed to the way that everybody else has socialized it before. There is a lot of, I would say, fear that mRNA goes back and it changes your DNA and things like that. But actually, the way that we like to think of it, and this is fundamental for us, is we think of it as code. So just as computers operate off of a binary system, zeros and ones, life actually sort of operates on a quaternary system, right? It's your nucleic acids, it's the AGCT. In the case of course, mRNA, you're looking at U instead. But that concept is basically enabling you to think of mRNA as an information molecule. And when you start to pull that apart and really think about that differently, what does that mean for medicines? So for us, we think of your DNA in its most basic sense, to take that computer analogy forward, as it's the storage. It's your main operating system, right? It's what my iPhone runs off of as an OS. That stuff doesn't change. That stuff we keep the same. It's your basic blueprints. What mRNA is essentially like those little apps that we're going to make. It is the codes for every single app on your iPhone. And each app has a different function. Each app represents potentially a different medicine, a different way that we can think about things. Fundamental to all of that, of course, is that it's code. And if you really understand the code and how to change it, what are the parameters that you need to work with to figure out how much mRNA is going to be translated by a cell? Can I change that? Can I translate that to the point where I can get dose response curves from a pharmacokinetic perspective? Well, that opens up different worlds. Can I deliver it in different ways to different tissues? That opens up different worlds. So once you start to understand the fundamentals of what mRNA really is and you think about it like code, well, then you can start to approach this from a platform perspective. And what does that mean? As a platform, what that means is essentially, it doesn't matter what the target is anymore. It's about understanding that platform and how I can change it. Because code is easy. If I understand it, I can write it. And if I can deliver it effectively, well, then I can change the types of medicines that I want to make, and I could do that in a very fast way. There is living examples now of how we can do that and unleashing that platform as a technology. The first, of course, is COVID. The virus has changed. The virus that we see now circulating in the population is not the same virus that started. It has evolved continuously. But we have, because of the way the platform works, can keep up with that. We can anticipate that. The goal is to be able to get ahead of it. Right? So the question is, what does the platform enable? It enables speed, the development of speed. The iterative development is the other part. That every time I make something, I can learn from it. And if I've set up my company and the structures accordingly very well, I can learn fast. I can make those changes. I can update that code, and I can deliver that very quickly. And that's the goal. That's the why Moderna actually doesn't think of itself as a pharma company. It is, obviously, it makes medicines, but it doesn't think of itself that way. We are not built structurally, internally, to act like a pharma company. We're actually built structurally like an IT company, and that's our philosophy. We are designed to continuously learn and do better and to do it quickly. mRNA has the ability to actually deliver proteins, that translation from the mRNA into the protein molecule, to different ways. In a cellular format, it could be proteins that could be secreted. It could be proteins that are anchored to a cell membrane, thereby making that cell potentially a target, or it could even be delivering proteins internally. What does that translate that into is essentially different types of what we call modalities. Modalities are essentially a combination of how do we deliver those medicines? Oops, sorry. Too fast. How do we deliver those medicines to what are their potential targets, as well as what are their endpoints? So effectively, different modalities exist for therapeutic targets. Different modalities exist for vaccine targets. Different ones and modalities are anchored in oncology for cancer, for example. Some are anchored in rare diseases. And in fact, that's what we are building. It's that combination of mRNA science, the delivery mechanisms of that mRNA, which vary, as well as how you make it. Can't underestimate the importance of the manufacturing, because that is actually how a lot of this magic actually comes together. Put together, and I've just listed a couple of proofs of concept here, is what those targets actually look like. And those targets are actually because there are clinical trial results associated with all of these. So at the top is the vaccines. With COVID, we feel like we've significantly understood the impact that we can have in infectious diseases. So we're heavy into infectious disease now. You will see products coming out, not just for COVID, but next, we've already filed for RSV. The next one is flu. We think we could do better than the existing flu vaccines. The existing flu vaccines are based on a technology where we grow up the viruses in eggs. It takes about a year in terms of actually the prediction of that structure, or what is the circulating virus, to the time that we can actually manufacture. We can condense that down to about 3 months now. If in fact we can do this, in theory, you can get better results. I'm going to switch over to oncology, and specifically on something called individualized neoantigen therapies, which is a very, very complicated term for a personalized cancer vaccine. Essentially, what we do is we take a tumor out of a cancer patient. In this case, we've done the trials in melanoma. Why? Because melanoma is actually a very dirty type of cancer. You get to see a lot of different types within it. Take the tumor, take that back to the lab, do an analysis of the tumor, do an analysis of your regular blood cells. Understand the difference genetically of what the markers are that is unique to your tumor and potentially antigenic, meaning it will elicit an immune response, and then use that to train your immune system to target your own cancer. That has actually gone through phase 2 trials. The results were actually quite remarkable. It used in conjunction with one of the standard therapies called Keytruda. It actually doubled the effect. So it's now been fast-tracked into phase 3, and I'll come back to this one in a moment because that means we have to build differently from a manufacturing standpoint going forward. The last one is in rare diseases. A lot of these things have been tackled or tried to tackle from a genetics therapy perspective. The approach with mRNA is if you understand the mutation in the particular enzyme, can I just supplement that? What's unique about these is that unlike a vaccine where you're giving an mRNA injection of a small amount and you want to elicit immune response, now I have to deliver at a therapeutic level an mRNA molecule over a course of time, so a larger volume, to produce that molecule in your body, and I have to do that without eliciting an immune response. It's a very different biological type of target to go for. The key to all of these things is no longer that medicines are made by chemicals or synthesizing those things, and then you metabolize those to get an effect. Now we've turned your body into the manufacturing plant of your own medicine. And that's the principle of the actual platform. And that's why we think you're sitting on the verge of something that's going to change medicine in very, very different ways. The way I explained this early on in the pandemic to my neighbor 6 feet across the driveway was it's like we've just invented surgery. It will change so many different things about medicine. So, what are we doing? Besides all of that, there were a couple of things that I did want to highlight because I know there are interests from a global public health perspective here in this room. The first is that the philosophy behind this technology and portions of what we want to do in infectious disease is that we don't want to ever see a pandemic do what it did. So how are we going to do this differently? And so we've actually set very ambitious targets of working with the WHO and the CEPI lists of pathogens that are likely to cause a pandemic in the future, and have targeted 15 of those to be in clinical trial by 2025. So again, speaking to how fast this can work and how quickly and how determined the company is to actually impact on this. There are two other actually aspects which I won't go into in a lot of detail, but in terms of manufacturing, there is a plan around this, of which Canada is actually an integral part. We are building a manufacturing plant here in Canada, in Quebec, and it is part of what's called the Resilience Program. The second is mRNA access. So this is leveraging, for those of you who don't use iPhones and like the Android kind of model, it's basically, it's open sourcing the technology. It's understanding that there is no way a company of 5,000 people can do all of this themselves. So how do you make the technology available to others to explore that? And so with the mRNA access program, in fact, what you can do is you can write, we'll produce the materials and, and support your research for you to explore. And if we like it, then we would love to be able to continue to work with you going forward. Okay. On the manufacturing site, this is currently what it looks like. The slide is actually already out of date because it is continuously evolving. But what I wanted to highlight was essentially that Canada will have a manufacturing site. Of course, the US has the site, the original site. We're also building in the UK. We've also built one in Australia. And there are plans actually to put one into Kenya as well. One of the things that we learned during the pandemic is the supply chains don't exist and those infrastructures are not clear there. And so how do you enable access to technologies and to have those products readily available? Got to build there. So we're going to switch gears a little bit and we're going to start talking a little bit more about what is careers and what is it like to work in Moderna. I put this up on there because this is actually fundamental to something that our executive committees have really spent a lot of time on, which is what made Moderna special. And how do we feel like our success, which was a small company of just a few hundred people, managed to pull off so much during an extraordinary time. And so they've put it down in terms of not just what are our core values and how do we recruit, what are we looking for in people. In fact, you actually have to take a behavioral test just as part of the interview process. But we're looking for bold, collaborative, a one-team approach. There are no egos in Moderna. You will see some of the smartest people we've ever met, but at the same time, we're all humble. Nobody's there to prove anything about themselves. It's about understanding what that technology is going to do, and we are united in that mission of that's what we have to bring out, and we have to get it to patients as quickly as possible. They actually have gone a bit further. They've done an analysis of what are the behaviors that they want to maintain going forward. And so, in fact, during an interview process, you get exposed to what are these, what we call the mindsets. And a lot of thought has actually gone behind some of those things. So those are the things that we're actually trying to champion inside of each individual. And, you know, I was once asked actually at another talk, you know, what makes Moderna special? And we actually feel it's our people. And it's the way that we work. And so capturing that and trying to make sure that that culture is maintained, even if we were 100 people to if we grow to 10,000 people, is fundamental to everything that we do. So let's switch gears now in terms of jobs. And I'm going to actually give you two examples of what the potential looks like. Because I'll be very honest with you, I am not a mathematics specialist. I love the potential of AI. I'm starting to learn it. If somebody in this audience could once at some point tell me the solution to what Move 37 really meant. I would really like to understand that. So anybody who's doing AI would love to talk to you afterwards. But this is one of the examples from a manufacturing standpoint where you need to think differently about what you're building. So at the onset, we had to go from a clinical stage company that had never made a dose batch more than 100,000 vials before to one that actually had to deliver billions of doses around the world. It was about scaling up. How do you scale up a technology? How do you manage the workflows for those things within a manufacturing plant? Now, change the case study. Now that we've seen the personalized cancer vaccine results, well, now I'm not about scaling up volumes. Now I have to make thousands and thousands of individual level mRNAs at a manufacturing footprint in a very different way. Right? And they can't mix. You can't get somebody else's personalized cancer vaccine. So what does the footprint look like? So exercises are now going on within the manufacturing facilities of mapping all of that out as they go into phase 3 and they're scaling up for thousands of patients. And what is that going to look like for the future? And I can tell you that a lot of work is going into using digital information to understand, how do I manage workflows? How do I optimize? Because our goal is to, from the time you take the blood sample and the tumor sample, to get you a medicine should be 6 weeks. If we can get it down to 6 weeks, that's ideal. And just remember that there is a patient waiting at the end of the day whose life depends on that. So every minute counts. So everything is about mapping out per the minute how much time each step is taking, where can we optimize. The second one is on the clinical trials perspective, and I'll reorient you back to that CEPI target of here's the 15 different pathogens that we're after. It's just a drop in the bucket. Of course, we're also looking into bacterial targets, and in fact, that's already started in clinical trials for Lyme disease. But the concept here and the thing that we wanted to highlight is we don't know all of it. There are no targets necessarily, or targets that we think work for all of these different types of diseases. So structural biology is really important. The mathematics behind all of this is very important. So all of this is part and parcel into what makes an effective product. But for us, it's not just about the product, it's about the solution. Address the disease, understand how does it transmit, what makes sense. Is it going to be a vaccine? Is it going to be a therapeutic approach? How would you deliver that? The interesting thing about the technology is that for the first time, we can start to really think about public health in a different way. Whereas vaccines in traditional, and I've grown up in vaccines as a career, public health and those vaccines are basically, they don't change. They haven't changed for many decades. For most of them, they don't, with the exception of flu. But now, with this concept of iterative design and continuously getting better, and the fact that you can make these things so fast, well, now you can start to enable the ideas of precision public health. Much the way that we have precision medicine, maybe I don't need to actually vaccinate an entire population. Maybe I could achieve the same results, or even better, if I can effectively sub-vaccinate certain populations that are either highly more susceptible or more transmittable. Right? So it starts to, the technology is starting just to enable that thought pattern of what can we do differently. And if you take into the social factors of what we've experienced in this last pandemic, I think Pandora's box has been opened in the sense that nobody is going to willingly just take vaccines because the government told them to anymore. Right? We've seen what vaccine hesitancy is going to do. So we need to change. We need to think about this differently. And that's for your great minds to keep working on. So Moderna itself actually really loves digitizing everything. In fact, that's actually one of those mindsets in the early slides. We focus heavily on data and capturing that data. We automate everything. The reason why we can actually manufacture things within a span of like 3 weeks is because I can walk up to the computer terminal, I can put in the code that I want, and within 3 to 4 weeks, I will have clinical batches made in a completely automated suite, ready to go into trial. And that's because It's a new company. It didn't have legacy systems that it had to deal with. So it built with this mindset of build everything digitally first. And we continue that forward. So we're very much into AI. We've developed our own AI systems internally. We've adopted them, which is what the slide is showing. And we're doing some very innovative partnerships as well to look into sort of the next generations of what AI will look like and how can we get that for analyzing what future medicines could be. The last slide is basically, I had a chance to talk to one group, the clinical epidemiology group, and get their insights into what they felt would be jobs of the future. And I will caveat this by saying these are kind of the immediate jobs, because I think we can all agree that not everybody understands exactly what AI is going to do and take us towards. But there was a very nice article actually that just came out in the New England Journal of Medicine, which tried to capture some of the learnings, but then also point out where we just don't know yet, right? And so I got their interpretations, and I wanted to share those with you because I thought it was actually a really interesting conversation about how we want to adopt these things, but where there are still hesitancies. And so those are gaps that I think are things that could inform you about where you might want to play and where you see yourself. The job titles generally are data scientists, biostatisticians, and lab programmers. They sit within the clinical group or within an epidemiology group. There were two aspects that they looked at. One is, and two trends to note. The first is that regulatory authorities around the world are now more and more interested in understanding real-world evidence. So outside of a clinical setting, where you do a very interventional-based trial under controlled settings, now we want to see how products are working in an actual population and learn from that, right? Because that's valuable information as well. All too often, we've seen that clinical trials don't always give us everything, especially if they're very rare events. And so we don't find out about it until you put it into a population. So regulators are also starting to want to see more and more of that data. So understanding some of those things like confounding variables, and I saw some people's research here just as I was setting in, was looking at these types of things, especially the societal factors. I think there's a need and an expertise around study design going forward in those large data sets, complex data sets with inherent biases, and how to remove those biases. You know, a great example that was given to me, and I jotted down some notes there, was essentially that when we're looking now at the COVID vaccine effectiveness studies, one of the great data sources is the Kaiser Permanente source in the US, which in itself is an inherently biased data set, because it's an insurance-based data set, so only certain people qualify, right? But yet, it's a large linked network, so a very valuable tool to look into. But when you start to dig deeper into a lot of these things, we want to understand, well, who is high risk going forward? And you start to understand more about human behavior. Those who are high risk tend to be more proactive in getting vaccinated. But those who aren't, and don't see themselves as high risk, don't tend to be more proactive in getting vaccinated. So when I generate results, well, how do I remove the bias from those effectiveness studies, knowing that those people who are high risk and have more complicated medical backgrounds are also the ones where I'm showing effectiveness in, which we want to see, but then is that truly the value or the true value of what the effectiveness of the vaccine really is for the broader population? We don't know. You have to be able to tease those details apart, understanding human behavior as well. The other one is in the clinical trial aspect, of which there has been a lot of talk about how AI can really help in that regard. The conversation that we're having is that it's interesting. Clinical trials are very hypothesis driven. You put in an intervention and you test it. It's very much based on the scientific method in that respect. AI actually takes a very different approach. You know, it's not necessarily hypothesis driven. It's actually there to generate hypotheses in many respects. And so it takes different types of math. And when we've tested it in some respects, you do start to see that the system itself is actually quite susceptible to different biases as well. In fact, one case example that was mentioned to me was that interestingly enough, it actually made up its own references, and they and you can't have that, because from a regulatory perspective, reproducibility of results is one of those fundamental things that we have to have, and AI hasn't yet been able to do that. So where this is going, it means that you stand on the cusp of a lot of changes where we see this as a powerful tool going forward. Math is a great, powerful tool, but we have to develop new ways and understandings and approaches for this. So with that, I'm actually going to stop, and I think we're at time actually. So, but I'm happy. I'll be around for the next little while, and I'm happy to take questions as well.
So raise your hand if you have any questions for Shehzad.
Just no heavy math questions, honestly. Like, I'm an immunologist. I don't do math.
I just wanted to say excellent presentation. There's so many things, like I heard of mRNA, but I didn't exactly knew. So this one kind of summed up. So thank you for that. And I was just interested, it's not a technical question, just in the cancer, that sounded like I wasn't aware of that too. So you know what, it's still in the trial phase, right?
Yes.
Or okay, what kind of cancers are?
So they're starting off with phase 3 for melanoma, but then they're expanding into other solid tumors as well.
Okay.
A lot of this is just an active discussion, both with Merck as our partner for some of this, but then there's an entire oncology division now that's growing for other oncology targets in general. One of the things that you can think about is, actually, I'll just say, one of the things early on that actually got me very excited and why I decided to go to Moderna wasn't necessarily the COVID results, even though for me personally, I've been in vaccines, and when I saw the results, I knew that's where I wanted to be. But one of the things that they had shown was in their, one of the infectious disease programs, they had actually created monoclonal antibodies using mRNA, specific monoclonal antibodies. So if you know the structure of an antibody, it's a heavy chain and a light chain. So you use one mRNA to create a heavy chain, one mRNA to make a light chain. And inside the cell, it actually will assemble into the form of a Y, which is the antibody. And we can make it very specific. And I can then deliver that to a therapeutic dose. I can show function in clinical trial, and I can show dose response. Think of all the monoclonal antibody therapies out there right now. They are made in exogenous systems where you have to manufacture those humanized monoclonal antibodies, then somebody comes in for an IV delivery. Now your body can make it. I just give it the instructions. It's set to disrupt everything about that therapeutic area. And to me, those are the most expensive drugs in the world. You look at the top 10 selling drugs, Remicade, all of them, they're all monoclonal antibodies. The doses are very expensive. I'm in medical. I will not make comments about what charges there will be, but the fact that you can now do this differently will change things, right? And that was exciting to me. So from a cancer perspective, Keytruda is a PD-L1 target, right? So there are different targets from an immune system perspective that if you can make those monoclonal antibodies for, it opens up a world of possibilities for oncology targets and others, other immune-caused or chronic diseases as well. Saw a hand back there.
I can't wait to watch the movie.
I feel a bit cheaty because I've got two questions. So
right.
The first one is kind of with Moderna Canada, what kind of collaborations have you formed with academic institutions or the public health institutions, and what kind of projects have you been working, those collaborations been about?
Sure, so actually, there's three sort of buckets that we think about. There's basic R&D, things about improving mRNA. There are other types of RNA out there as well, not just messenger RNA. It's the delivery mechanisms, so lipid nanoparticles, for example, which we didn't touch on in this presentation. But if you want to talk about how do you target different tissues, it comes down to the lipid nanoparticles, for which we have programs in all of this, and a battery of different LNPs for short, that help us to say target the liver for certain types of diseases or the lungs, so on and so forth. So continuing those types of collaborations, of which a couple I can mention. So we have blanket agreements now set up with the University of Toronto as well as McGill. We also have individual sort of R&D projects that are going on at other universities across Canada, and we're actively continuing to look. And so that's one area. Second area is in clinical trials themselves. When I started, we didn't have any clinical trials besides the ones in the US. The first clinical trial that we brought up here was the Kids Cove trial, which is when the COVID vaccine started to expand beyond just adults and into children, of which we got over 10 clinical trial sites here in Canada spread out across the provinces. We are now at 11 clinical trials in Canada, not just for COVID, but for other infectious diseases, as well as for rare diseases. So we actually have 3 different rare disease trials going on in Canada right now at a few institutions. And the next target is oncology. I really want to get oncology trials up here as well. The third bucket is in the epidemiology surveillance real world space, which in a safe space, I will say, is a challenge in Canada. We're determined. The reason why is because, and this one is a personal mission for me, is we're a public health system. As I mentioned, a lot of the data that has been driven so far has been through inherently biased systems, especially in the US. Imagine unlocking the power of what the public health system means, where I can capture somebody from birth till death, where I know what prescriptions they take, and so on and so forth. It gets into those areas around data privacy 100%. But those things have been worked out, and this is the argument that we've made, is Europe has done this already in many different ways. Why can't we figure ourselves out? We can't get our provinces to talk with data between those data provinces. So we need to get on that. And so there is work that's going on to figure out how we can actually do this right. Because I think there's inherent value in those data systems if we can figure out a proper way of accessing and using them and doing this collaboratively. And that's the key part, is collaborative type research, because the science is evolving and we need to do this together. We can't do this in our separate spaces where it's transactional type research. That, I think, doesn't serve anybody very well because it slows things down. That's the goal. I don't have any successes to tell you yet.
The other one goes back to your answer to your previous question. You kind of mentioned constructing antibodies for 2 different sides of the light and the heavy chain for antibodies for monoclonal antibody treatment. Has there been any kind of thought of using that to produce rapid antigen tests? So I know from previous work on gonorrhea that there's huge issues with, if you could test someone within 20 minutes using like COVID type rapid antigen tests for a specific, say if they've got something that might be antibiotic resistant to a specific drug, that would be like, you could give them the treatment there and then, and not just wait to grow it in a lab. Yeah, so it's a great question. The current thinking within Moderna is we're not making diagnostics. So I don't know which way we'll grow and how we'll grow. I've been asked the same type of question of would you get into animal vaccines and things like that. We're trying to stay focused right now in terms of our growth and what we need to do. But I think that's open for others to figure out how to do.
Okay, thank you very much for your presentation, and I appreciate that. I want to ask concerning the neglected tropical diseases, because I'm coming from Africa. So when you talk about you have a collaboration in Kenya, so I want to see what kind of job opportunities is there, because I know that neglected tropical disease is one of those diseases that we are most of us from Africa are suffering. So most of them doesn't have vaccines. So talking about developing a vaccine for that for me is a very good thing. But I want to see, as a mathematician, what kind of job opportunity can someone have there? Especially if you are coming from Africa, maybe you want to work somewhere like in Kenya where you have a collaboration. What kind of job is there? It's an opportunity that is there. So that's just what I want to ask. Thank you.
Yeah, no, it's a good question. I would say it's very early days, so it's basically they're still conceptualizing what they're going to put in Kenya. I know it's manufacturing based, so a lot of the jobs would be from a manufacturing perspective. For anybody who has ever worked on the manufacturing side, there's an entire analytical sciences and bioprocess development and R&D side as well that requires good R&D in science, because that's actually the bottleneck of producing a lot of medicines. It's actually quite easy to find targets. It's hard to find targets that are stable or that actually work. And that's the traditional part that that's why you see a lot of trials failing eventually, is because it doesn't make a product at the end of the day. It may work, but in a lab setting or at a very small scale, but that's not a product that eventually can go into people. And so that lesson and those learnings are actually something that as a company, everybody has to grow and understand how they're going to address. And with the new technology, there will be opportunities to figure that out. Beyond that, I can't actually give you more details at this point. It's very early days.
Hi, I have a quick question about Disease X, and in particular, how the iterative model can work for a disease that we don't know anything about yet, that could potentially cause like the next epidemic.
Yeah, great question. And I don't have an answer for you, unfortunately, on that one. That's going to be my answer at this point. Yeah.
All right. Thank you very much, Shehzad. And everybody give a round of applause for him.
Thank you.